Next on our list of organelles is the endoplasmic reticulum. The endoplasmic reticulum is a tremendous organelle in that it fills up a large portion of most cells. It's actually a network of flattened sacs of membrane and tubular structures connecting all of those sacs. The endoplasmic reticulum is actually connected to the nuclear envelope. So it's one big sort of mesh of membrane. There are two forms of endoplasmic reticulum, what we call rough and smooth. Rough endoplasmic reticulum got its name because under the microscope, the surface of that structure looks very rough. And the reason it looks rough is because it is studded with ribosomes. There are lots and lots of individual ribosomes that are sitting on the membrane surrounding the endoplasmic reticulum. Remember, the endoplasmic reticulum is a single membrane organelle. The rough ER, with its associated ribosomes, has the job of producing and modifying proteins, as we said a couple of minutes ago. The particular types of proteins that pass through the rough ER are proteins that are destined to live in a membrane somewhere, either in the cell membrane itself or in membrane around one of the organelles. The other types of proteins that tend to pass through the RER are proteins that are destined to be secreted from the cell. Now, the smooth ER got its name because it doesn't have any ribosomes associated with it, so it looks very smooth underneath the microscope. The smooth ER has nothing to do with protein synthesis. When you look at it under the microscope, it also looks different from the rough ER. It looks more tubular and less flattened than the RER does. There are three separate functions of the smooth endoplasmic reticulum. These include the synthesis of lipids, the detoxification of certain molecules, including certain drugs, and the storage and regulation of calcium. Calcium ions are used by cells for different types of communication. When we talk about skeletal muscle contraction, calcium will play a big role there. On this slide, you can see an artist's drawing of the endoplasmic reticulum, both the smooth and the rough. And the artist has cut through the endoplasmic reticulum so you can see these cut surfaces. You can also see this purple structure over here. This represents the nucleus. So remember, the ER is continuous with the nuclear envelope. The rough ER is actually attached to the outer membrane of the nuclear envelope. You can see that it's really a series of these flattened sacs of membrane. They look like pancakes. And there are tubes connecting the pancakes. So in other words, a molecule could travel out a nuclear pore and into the RER, and then travel through this network and these connecting tubes all the way through the organelle. So it's one big network of membrane. And again, it looks rough because of these little ribosomes that are sitting all over its surface like little dots. Now the RER is connected to the SER, shown in orange in this drawing. I think you'll agree that the SER looks a lot more tubular and a lot less flattened than the RER does. Remember, the SER has nothing to do with protein, even though it's connected to the rough ER. The SER is about lipid synthesis and drug detoxification and calcium. On this slide, we're looking at two transmission electron microscope images of the rough ER here on the left and the smooth ER here on the right. If you look at this image on the left, you can really see the flattened sort of sac nature of the rough ER. And you can also see why someone looked at this under a microscope and described it as being rough. Each one of these flattened sacs of membrane, these little pancakes of membrane, is just covered with these little black dots. Those are the ribosomes. So all of this is rough ER. Now compare that to the image on the right. In this image, you can see a portion of the nucleus right here. And then you can see rough ER here and smooth ER here. Now I know this image isn't as clear as the other one. I know that you can't see the sacs of the RER very well here, but I think you can see that it's covered in these little black dots. That's rough endoplasmic reticulum. Compare that to this side. You can still see the little sacs of membrane, although there are a lot more sort of circular regions. It's more tubular than the RER is. And there are no black dots. There are no ribosomes here. Rough ER and smooth ER under the electron microscope. Nearby to the endoplasmic reticulum is an organelle called the Golgi apparatus. The Golgi has gone through a few name changes over the past generation or two. It used to be called the Golgi body, but now it's more commonly called the Golgi apparatus. Like the rough ER, the Golgi apparatus is composed of flattened sacs of membrane that are stacked on top of each other like pancakes. But there are a couple of structural features that make it very different from the RER. First of all, the individual sacs of the Golgi apparatus are not attached to each other, and they're not attached to the ER. So the Golgi is completely separate from the ER, and each of the sacs is separate from the other sacs. Now, there are two distinct sides to the Golgi. In other words, we describe it based on its location in the cell. There's what we call the cis Golgi. That's the side of the Golgi apparatus that faces the ER. The opposite side is called the trans Golgi, and that's the side that faces away from the ER toward the cell membrane or the plasma membrane. Now, what's happening inside the Golgi apparatus largely has to do with carbohydrate. There are some complex carbohydrates, for example, that get synthesized inside the Golgi apparatus. But there are also some proteins that will come to the Golgi apparatus in order to be further modified. And the modification that happens inside the Golgi is with carbohydrate. In other words, bits of carbohydrate, short chains of carbohydrate, get added on to some proteins, and that changes the chemical nature of that protein. It becomes what we call a glycoprotein at that point. Now, in order for things to travel from one sac to another, through the Golgi apparatus. Since the sacs aren't connected to each other, we use what are called transport vesicles. Remember, these are small circular organelles with a single layer of membrane, and they're basically empty compartments. The materials that are made in the Golgi apparatus get packaged into these transport vesicles. The transport vesicle basically buds off of the surface of the flattened sac of membrane, and then it travels over to the next sac and it merges in with the membrane there, releasing the contents into that sac. Once material has passed all the way through the Golgi apparatus, it's also packaged into transport vesicles so it can be sent to its final destination in the cell. On this slide, you're looking at a drawing of the Golgi apparatus on the left and an EM image of the Golgi apparatus on the right. Let's look on the left first. You can see a portion of the nucleus up at the top in purple. The artist has then drawn in the rough endoplasmic reticulum. We don't see the smooth endoplasmic reticulum in this drawing. Down below, not attached to it, is the Golgi apparatus. The Golgi is made out of individual sacs of membrane that are stacked on each other, and it really does have this particular shape. In other words, the sacs get larger and larger and then smaller and smaller as you go from one side to the next. Remember, this is referred to as the cis Golgi. This is the side that faces the ER. This is referred to as the trans Golgi. This is the side that faces the cell membrane. What's happening in this organelle is the production of carbohydrate, particularly complex carbohydrate, and the modification of proteins. You can see that there's a little circular organelle right here. This is a transport vesicle, and it's carrying a protein from the ER into the Golgi. The membrane around the transport vesicle is going to attach to and merge with the Golgi membrane, and the protein will be released into the Golgi apparatus. It's going to get modified as it travels through each region of the Golgi, and that's what they're showing us here. This little peanut-shaped yellow structure represents the protein, and I think you can see that there's a short chain, a line added on to that protein. That represents a piece of carbohydrate. Once the modification is complete, the protein will be packaged into another transport vesicle and it will be sent out to wherever its final destination is. In this drawing, the final destination is outside of the cell. This protein is going to be secreted from the cell. If you look over here in the EM image, this is the Golgi apparatus. Now again, this doesn't look like the perfect drawing that we see on the left, but you can see a series of sacs that are laying on top of each other. You can also see a bunch of circular, very small circular organelles surrounding this stack. Again, depending on how the cell is oriented on the microscope slide, the Golgi is going to look slightly different from image to image to image. You can't see the nucleus in this image. You can see a tiny portion of ER over here. So this would be the cis face or the cis Golgi. This would be the trans face. And the cell membrane would be over here somewhere. Now, one other thing I want to mention before we leave the organelles, and that's this. When you look under a microscope at a cell, you can often distinguish the nucleus, the rough ER, the smooth ER, the Golgi apparatus, the mitochondria, but it's very difficult to distinguish the small circular organelles. The lysosome, the peroxisome, and the transport vesicle are all small circular organelles. You have to be quite expert at EM imagery in order to distinguish those three structures from each other under the microscope. Let's move on now and talk about some of the structural features of the plasma membrane. Remember that the plasma membrane, as well as the membranes surrounding the organelles, are a bilayer of material, and they're made primarily from phospholipids. Now, membranes are also full of proteins, and we'll talk about membrane proteins in just a little bit. But right now, let's focus on the structure of these phospholipids. On this slide, in the drawing at the bottom, you're looking at an artist's depiction of an individual phospholipid molecule. Remember, phospholipids are one of the three types of lipid that you find in a living cell. There are phospholipids, there are fats or triglycerides, and there are sterols. Phospholipids make up membranes. Structurally, each individual phospholipid is composed of a head region, what's drawn as a circle, and two tails coming off of the head. One head and two tails, therefore, make up one phospholipid. When you bring a whole bunch of phospholipid molecules together, they organize themselves like this. You can see this is a two-layered structure, so we refer to it as a bilayer. And this is a membrane. The phospholipids are not covalently bound to each other in a membrane. They're simply attracted to each other based on their chemistry, and that's what holds them together. The head region of a phospholipid is composed of two things: a molecule of glycerol and a phosphate group. No, it's difficult to see, but if we enlarge this, you can see this structure right here. This is glycerol. And this structure right here, this is a phosphate group. And together, those two things make up the head. Now, glycerol and phosphate are both polar. So the head region of a phospholipid is polar and therefore hydrophilic. The two tails are very different. The artist has drawn these tails as containing these zigzag lines. This represents lots and lots of carbon and hydrogen, or what we would call in chemistry a hydrocarbon. The hydrocarbon is attached right here to the head at what we call a carboxyl group. So the hydrocarbon and the carboxyl group together create what we call a fatty acid. So the tails are made out of fatty acids, two of them. Fatty acids, because they contain so much carbon and hydrogen, are nonpolar and therefore hydrophobic. One other thing to notice about these fatty acids: notice that one of them is quite straight and the other one has a very clear bend in it, or what we call a kink. The straight fatty acid is called saturated, It contains as much hydrogen as is chemically possible. The bent or kinked fatty acid is called unsaturated. It has slightly less hydrogen in it because of this. This double blue line represents a double bond between two carbon atoms. When you put double bonds inside of fatty acids, you cause them to kink like this. That's going to be important to the permeability of the membrane. You're probably aware of the fact that things like butter are saturated fats. Things like vegetable oil are unsaturated. Saturated fats tend to be solid at room temperature, right? They tend to be much less fluid than unsaturated fats. Unsaturated fatty acids create much more fluid fats and much more fluid phospholipids as well. Membrane fluidity will matter when we start talking about whether or not molecules have an easy time or a hard time passing across membranes. For now, just understand that fatty acids can be saturated or unsaturated based upon whether there's any of those carbon-to-carbon double bonds present. Now, when a group of phospholipids comes together, like this, they spontaneously form the bilayer that is a membrane. The heads of each phospholipid will gather together to face the outside of the cell and to face the inside of the cell. All of the fatty acid tails will cluster in the middle. This is what we refer to as a head region, and this is the tail region of a membrane. Remember, these individual phospholipids are not attached to each other with covalent bonds. They are simply attracted to each other. All of the heads stay together because they are all polar. All of the tails stay together because they're all nonpolar. Hydrogen bonds hold the heads together. Van der Waals interactions hold the tails together. Remember, living things are full of water. There's lots of water outside of cells, and there's lots of water inside of cells. So the polar and hydrophilic heads will orient themselves towards the water, both inside and outside the cell. The hydrophobic tails will stay away from the water, tucked in the middle. In addition to all of the phospholipids that make up a membrane, we also find lots and lots of protein. Remember, if you were to take a membrane and separate it out into its component parts, about half of the mass of the membrane would be phospholipid and about half would be protein. Membranes are packed with proteins. We can describe the proteins in a membrane based on their structure. There are what we call integral membrane proteins and peripheral membrane proteins. Now, integral membrane proteins, like the name suggests, are integrated into the phospholipids. Some integral membrane proteins only cross one of the layers of phospholipids, and some cross both layers of the phospholipids. If an integral membrane protein crosses both layers, we call it a transmembrane protein. These transmembrane proteins, in fact, all integral membrane proteins, are amphipathic. Part of the protein is polar, the part that hangs out with the heads of the membrane, and part of the protein is nonpolar, the part that hangs out in the tail region. Regardless of whether we're talking about an integral protein that crosses one layer or a transmembrane protein that crosses both layers, these integral membrane proteins have several different functions in the membranes of cells. Some of them function as channels, literal tunnels or pores through the membrane. Some of them function as carrier proteins. They're going to help some molecules get across the membrane. Some of them function as pump proteins. They're going to pump molecules across the membrane using ATP for energy. Some of them serve as receptors, receiving information from both outside and inside of the cell. And some of them are going to serve as cell identity markers, particularly on the outside of the cell. The other class of membrane proteins are called peripheral membrane proteins. Like the name suggests, these proteins are on the periphery of the membrane. They don't cross into the tail region. Instead, these polar proteins are going to be associated only with the polar heads of one or the other layers of the membrane. Peripheral proteins have a much shorter list of functions. They can function as receptors, and they can function as cell identity markers. Take a look at this drawing of a piece of cell membrane. You can see in the picture that it's just a little square section of the membrane that would cover the entire outside of a cell. You can see the phospholipids. They are drawn with these orange-red colored heads and these yellow colored fatty acid tails. In this picture, this represents the outside of the cell, and this represents the inside of the cell. So the heads on both sides of the membrane are facing the watery environment, and the tails are tucked out away from the water in the middle of the membrane. This is referred to as a bilayer. The proteins that are found in the membrane are drawn in blue. Some of them have been cut in half so we can look inside of them. You can see that some of these proteins cross one layer or the other. Some of them go all the way across both layers, and some of them sit only on one side or the other. So integral membrane proteins, transmembrane proteins, and peripheral proteins. Notice that the artist has drawn these proteins with different sizes and different shapes. Every protein has a unique three-dimensional shape that gives it its own identity, its own unique function. Notice too that some of these proteins have these strange looking branching structures attached to them, particularly on the outside surface of the membrane. These are what we would call glycoproteins. Remember, we mentioned those in relation to the Golgi apparatus. Glycoproteins are often involved in cell identity. Integral membrane proteins can serve a lot of functions. Some of them act as channels, some of them act as carriers. In both of those instances, the protein is helping something get across the membrane. We're going to talk about membrane transport in our next lecture, but for right now, I want you to look at how channel proteins and carrier proteins are structured. Up at the top of this slide, you're looking at a very small piece of membrane. The pink molecules represent the phospholipids, and this blue and green structure here represents a channel protein. It's been cut in half so we can look inside. Channel proteins are transmembrane proteins. They travel straight across both layers of the membrane. Remember, proteins are enormous molecules. They are macromolecules. Most of them contain hundreds or even thousands of individual amino acids. Each amino acid has its own chemistry. Some of them are polar, some of them are nonpolar. Some of them have charges, some of them don't. So if you were to take a big long string of amino acids and you were to fold it up to create a shape like this that has a tunnel through the middle, you would have to make sure that you grouped the amino acids according to the chemistry that would be needed. This is what I mean. In order for this protein to sit in this membrane, it has to be able to sit in the tail region. And the tail region is non-polar. So that means that the amino acids found on the outside surface of this channel also have to have non-polar chemistry. And that's what you're looking at in the darker color. In comparison, the amino acids on the inside of the channel would have to be polar. And that's because water is passing through this channel along with whatever molecule is coming across the membrane. Now take a look at the image at the bottom, which represents one of these carrier proteins. These are also transmembrane proteins, but instead of having a tunnel through the middle, they have an opening on one side of the membrane or the other. What happens with a carrier is that the molecule that needs to get across the membrane is going to come in and bind to the carrier in that opening. That binding of the molecule with the carrier is going to cause the carrier to change shape. And it will change the opening from this side of the membrane to the other side of the membrane. Pretty slick. That will allow the molecule to come off and into the inside of the cell. The action that you see with carrier proteins is a kind of a process that we will see again and again in physiological systems inside of cells. You can think of the molecule that is being carried across the membrane as a ligand or a ligand, depending on how you pronounce that term. That word is very generic. It just means a molecule, a specific molecule. It turns out that a lot of the physiology in the human body involves ligands binding to receptors, just like we see that molecule binding to the carrier protein. And just like we saw with the carrier protein, when a ligand binds to a receptor, it causes the receptor to change shape. Ligands fit receptors like a key fits in a lock. The binding site, the opening site on the protein, is going to fit the ligand perfectly, just like a key fits into a lock. As that ligand binds to the receptor, you're essentially creating a brand new molecule. Remember this: whenever an atom or a molecule binds to a protein, the protein is going to change shape. Each protein folds up in a very particular, very unique way. If you add something onto it, you're going to cause that shape to change. It will fold slightly differently because of that additional molecule. It's the change in the shape that will change the function of the protein and cause something to happen. So when a ligand, again, any particular molecule, binds to a receptor in the membrane of a cell, there will be a shape change that happens. And that shape change is going to trigger some action in the cell. This is a very common physiological process in living cells.